So thank you so much. First, I'd like to thank the organizers, IPM, for putting together this great event, and also Dr. Gavina for giving me the opportunity to present some of our work. So I think most of you obviously are aware of the space of 3D printing. So I will kind of cut to the chase. So today I want to kind of use my time and the stage here to share some of our effort in translating 3D printing and the opportunities we feel that exist in biomedicine, and also an area that perhaps is not something that people have touched upon is sustainability, and also what we think is a reality check of the challenges with 3D printing. So my talk is broken up into three parts, which I'm going to try to spend equal time on: progress in 3D printing towards clinical translation, and I'm going to specifically focus on example from our lab. Then I'm also going to use the next portion I'm going to talk about how 3D printing could play a role in developing a sustainable ecosystem. And then finally, I'm going to conclude my talk with some thoughts on limitation and challenges in fully exploiting 3D bioprinting. So this is just a collage of information that basically says there's a huge commercial opportunity here, which is there's hardware development and bioinks, which we are very much interested in. Then there's a lot of other biology stuff that needs to be figured out, but there's a commercial opportunity here. So last year we wrote a review in Advanced Sciences where we tried to reframe this discussion, and we identified bioinks as like an extremely critical component for the future of translational bioprinting. And the truth is, there is really no clinically let's say, approved product that has come out of bioprinting. If you would like to call printing of polycaprolactone as bioprinting, which I don't think it is a thermoplastic polymer, there is really no product out there that combines bioink and cells to actually to make something clinically meaningful. And so we identified a couple of issues that we need to be able to develop inks that can be GMP compliant and should be amenable for standardization, which we heard also from other speakers. So we kind of developed a framework for translatable bioinks that includes very rigorous characterization and also a lot of extensive testing to understand the impact of the bioink and the biology, which people do not do. They just show a lot of green colored cells. That means nothing. You really don't know what has happened to the cells after you process them. And so this kind of is broken down to different levels of translation, and you could kind of represent the same thing into a translational pipeline and try to identify where do your bioinks fall in this translational pipeline. So what I'm going to share today with you essentially is the progress we made until TR level 4, and we are essentially focusing on trying to get it past that. So the first example I would like to share with you is an example on bone repair. Bone is actually one of the most extensively studied tissue in terms of regenerative potential. Is also where biomaterials have seen a lot of application. So when you think about bone repair, you can think about bone formation biologically being like two different ways. One is what is called direct bone formation from mesenchymal stem cells, which happens during embryonic development. And in the adult, it's mainly this process called endochondral ossification. So our goal was to see how we can develop new ideas in biology, in engineering, and also in combining these two and processing to come up with a solution. And so this essentially is a confluence of several disciplines: material science, engineering, biology. And since much of our effort is inspired by development, we call this developmental engineering. It's not a new idea. It's been around for a while, and we would like to bring this into bioprinting. So the ink that we use is this carboxylated agarose bioink, which is patented and which is being developed by our lab, and also will soon receive CE marking. So that means whatever technology we develop using this particular material can actually enter the clinic. So to kind of gain access to this, we kind of leaned upon some of our earlier work we had done in understanding fundamentally the biological processes involved in the formation of cartilage and bone from human mesenchymal cells, which we obtained from human donors. And we identified that this aggregation step how MSCs come together, which are mesenchymal stem cells, actually triggers an endogenous program of chondrogenesis that people did not know before existed in these cells, and that allows us to essentially use this aggregation phenomena as a way to trigger the biological process. And so to get to that, we combine this element 1 with element 2, which is we have developed these inks that can be printed into all sorts of structures without the need for a support structure. And they're mechanically stable. That means we can print them with very, very high concentration of cells, like 100 million cells per ml. And then these implants can be implanted and they will retain their shape. And this is very important if you want to prescribe a shape to a material before you implant. And so here you see different shapes we have realized, including complex toroid shapes, a complete human mandible that has been printed down to scale, where you can also see the missing tooth. And so using this system, what we were able to show is that we can engineer what is called an hypertrophic chondrocyte template, which is a stage that is biologically found, which is in vivo. When you look at how bones develop in a developing, like a child as they go through adulthood, is the formation of cartilage that slowly becomes bone. So actually, a child when it's born has very little bone in the anatomy. It's mainly cartilage. So the cartilage has to be a specific phenotype, which is called hypertrophic chondrocytes. So what we have shown is we can take these inks and we can load them up with human mesenchymal cells. And if we design the ink properly, these cells will spontaneously aggregate, and they will aggregate into certain size of clusters that very favorably drives this chondrogenesis. And as a result, we can get these templates of tissue that are basically made up of hypertrophic chondrocytes. And so when we implant them essentially into the in the human body, we end up getting bone tissue that's exactly the shape that we had prescribed ex vivo. So this is something which we are now trying to bring it to the clinic, and we're developing some of the know-how to do that, and especially in terms of how do we implement such a process in a biofabrication workflow that is compatible with clinical application. So we are essentially somewhere between TRL4 and TRL5. So there are also, you heard a lot today about thermoplastics, especially PEEK, and I just thought I'd share with you some of the recent things that I found. There's a lot of translational efforts in bringing PEEK printed, 3D printed materials into the clinic. And as early as 2018, there was a report of a clavicle bone replacement using 3D printing out of China. And then there were several, then there's been a clinical trial, which I'm not sure what the status of it is right now, where they use 3D printed PEEK cranioplasty implant based on the Evonik, the PEEK that's produced by Evonik, and the Appium printer, which is currently ongoing. And the last thing I know is they've recruited about 40 patients to take the trial to the next level. There have also been reports of PEEK implants for repairing mandibular defects. And recently, there was an FDA 510K clearance for the Curateva FFD-printed PEEK intravertebral spinal fusion products. So all in all, we can say that like 3D printing is sort of coming of age, and hopefully soon we will be able to see something in the clinic. So at this point, I would like to kind of switch gears and kind of introduce you to sustainability. I'm going to throw some numbers on the screen. 115 million tons. Which is a lot of, which actually is several times the weight of the human mass on the planet. 8 billion people, which is the number of people on the planet. And 3% of US energy consumption, which is 12% of US industrial energy use. What am I talking about? I'm talking about single-use plastic. So when you think of single-use plastic, and you wonder where does 3D printing play a role in this, when you think of single-use plastic, what comes to mind is essentially bottles. And one single, like plastic bag, for example, is equivalent to 1.5 kilograms of carbon dioxide, or it's like driving a car for 8 kilometers, or an emission of almost 1 cubic meter of carbon dioxide. So this picture kind of says it all. We generate a lot of plastic. But I think there is a dirty story which has not been addressed, is the generation of plastic and the environmental impact of it. And what is the role of our own biological research in this area? So if you think about single-use plastics, you can see it's very energy intensive, it's water intensive, it depletes natural resources, and its global consumption is not slowing down, it's growing up. And in fact, biomedical research is one of the biggest contributors of single-use plastic. almost everything we use in the lab, if we use it for barely a couple of seconds and we throw it out. And we don't think about it. And the COVID pandemic just highlighted this, how much plastic waste we can generate. And nobody wants to pick this up, nobody wants to deal with it because it's contaminated in some way. And they all end up in some landfill. So we in our lab are very conscious about it. And so we started thinking about it. I'm going to share one example that, in interest of time, is if you think about cancer biology, there's a lot of single-use plastic in cancer biology research. It starts off with this first step when we grow the cells in plastic dishes or like flasks. And then you assemble these cells to create spheroids. And they're typically a system called a hanging drop system. And just for people to get an idea, this system here produced by Perfecta costs about like $200, $300 a plate. We use it once and you throw it. And then if you look at it, there's a lot of time and manpower involved. And then if you bring in other technologies such as microfluidics, this whole thing is basically time and cost intensive and uses a lot of plastic. So our goal was, can we just do a simple thing, nothing big dramatic? We go through a lot of these plates. Can we produce this plate by ourselves? So we have actually introduced a customized hanging drop inserts we call Q-serts, which as opposed to the Perfecta plate, which is an array of 96 plates, these are essentially clover-shaped 4x4 valves which can be inserted. They can be color-coded so you know what your experiments are. And you can essentially assemble as many as you want in a plate. So if you just want half the plate, you can just assemble half of these inserts and thereby be able to reduce the amount of plastic we use. And since it is produced using PLA and we have developed all the hardware for it, the code for it and everything, so essentially we can recycle this, make the filament back, and print the new plates in our lab. And so we have actually validated these plates using automation, using robotics, like using a pipetting robot. And I've found that, in fact, the output is the same as you would if you were to spend on an expensive plate that cannot be recycled. So you may not realize, but even a simple step like this could have a huge impact on how plastics are consumed in the lab. So we have also kind of pushed the 3D printing envelope to actually use environmentally sourced carbon-negative polymers as a way of producing materials that exceed the performance of synthetic plastics. And this is a work we published last year, where we were able to take polysaccharides which are produced by algae, in a couple of different polysaccharides. One of them is an agarose-modified polysaccharide, and the other one is alginate. And by developing this engineering paradigm, so what you can see here is an Ashby plot, which is a plot of material properties like modulus versus ultimate strength. And you can see where all the different materials fall. And what you see here is the gray zone here in the middle is these materials we have developed, which actually perform like plastic, but they're completely compostable. And what is more interesting is we can actually process these materials using 3D printing. So what you see here is an example where we have taken these materials. They are soft materials, they're like hydrogels. So you can print them into sheets. As you see here, these are dried sheets and a wet sheet. There are 3 layers of sheets that have been printed on top of each other. They can be printed using normal 3D printing, like device that's found in the lab, which is scalable from a commercial standpoint. And more interestingly, we can print these kind of patterns on wooden kind of laminates and create a glue that is a completely biologically friendly glue that we can use to glue wooden laminates together, and you can then dissociate the laminates by just putting them in hot water. So this is an idea that we are really excited about, because the application of 3D printing is not just limited to medical devices, but we believe we'll go beyond it to products that are used in the biomedical space, including everything from labware to other kind of coatings on cell culture devices and such. So to get to my last part of my talk, which I have about 5 minutes left, so you heard a lot about printing, about all kinds of printing. But when we got into this field and we started using fused filament deposition or fused filament modeling or whatever you want to call it, we realized that it's a diverse ecosystem of hardware. I mean, you can buy so many different printers and you don't know which one is good, which one is going to perform well, and which is not going to perform well. Or even more bigger question is, will that company exist tomorrow? So a lot of the work that is done in the lab, we use printers and we develop workflow around printers, and we realize maybe the printer company may not be around when we want to build on it. So this is one thing I've identified as a limitation and challenge, which I thought maybe I should share with you, because this is something Irina mentioned that could you kind of tune your talk in that direction. Then what we've noticed, there are very few systems that are qualified. You can think about 3D systems have their printer. The Appium is another company that has printers. Then the fact that many of these printers use proprietary software and hardware, that means it's very difficult for you to know why something went well and why something went wrong. And this is something we have grappled with ourselves. And the question is also, are these scalable? I mean, of course, we have seen examples of printers that are like are compatible with point of care, but if you think about them, they are not cheap. You cannot expect a lab to be able to buy a $150,000 printer. So we talk about innovation, and I think a lot of times we forget much of the innovation happens in academia, and in order for this to happen, we need to be able to make these more accessible. Then standardization, we just heard the speaker earlier talk about standardization. And of course, the solution they had was to develop a printer. But I kind of, maybe I'm going to take a different tack on it. It's kind of, when you already have a printer, why would I want to go buy another printer? So there are a lot of people with bioprinters, but what is missing is a way to compare data that we generate. That means a standardization framework where I can see, look, what I have printed versus you, can I compare the output? Can I compare if these two outputs are comparable? So this is to everything from, if you think about fused filament deposition, we're talking delamination, fracture, batch-to-batch variation. Then we think about safety of the filament that we buy. Is it actually compatible? Is it free of additives? Can we get access to virgin materials of high quality and a reliable quality assurance information? Then there's a cost here, and I'm just going to go through this very quickly. Like printers are not cheap. They start off sometimes being very cheap, but the cheap ones don't perform sometimes as well as expensive, but doesn't mean expensive ones are better. The infrastructure to actually get something to clinic is very, very cost prohibitive, and not everybody can implement it, so there's a lot of planning and regulatory compliance issues you have to go after. When you think about biology research, our own experiences, we all know it's time consuming. Then when you add on that the variability that bioprinting brings into it, you realize that you're compounding this problem even more. Then training of lab individuals, having well-trained workforce who can actually do quality work in the lab. And this is something I found is a single most thing. When you look at the quality of papers and publication, is the quality of the science is very poor, because there are people who are working on these projects do not have the appropriate training to function in this complex ecosystem. The factors impacting economics, like if you think about the wastage that is associated with 3D printing, which we don't talk about, all the trimmings and everything that cannot be recycled. And then, of course, printing one object at a time is going to lead to nowhere. How do you mass produce it? There's an economic and social impact that I do not have to further emphasize. So I feel like focused degree programs, there is new job opportunities for pan-disciplinary kind of interaction. There's definitely going to be improvement of quality of health through innovation, notably in cancer and inflammatory conditions. And we believe, there was a speaker earlier this morning spoke about how to print tablets. We do believe that the cost of drug development and pharma is going to come down a lot by pharma. So this is a economic and social opportunity. And of course, the reduced use of animals is something many of us feel passionate about. So to kind of finish my talk, I just want to kind of mention that there is a carbon footprint that is associated with 3D printing that we seldom talk about. Just to give you an example, if you want to print 3 times 10 millimeter size objects, and if you just use a material that needs to be melted at a high temperature, that corresponds to a significant increase in not only energy consumption, but CO2 production. And you might not think about it initially, but when you think about mass production, this is a very significant number, because you have to keep the printer heated, you have to maintain it hot when it's done, and then there's a cooling period, there's a lot of energy involved. So the simple question is 3D printing eco-friendly and sustainable? Maybe the answer right now is not quite, and this is something we want all of us, all the people in the field, to be aware of it. So finally, just as an outlook, the importance of academic industry partnerships cannot be overlooked. So that means addressing the shortage of well-trained and technical workforce, early tech exchange, academics usually focus on research grants, industry usually focus on can we make money, so we need to meet somewhere in between, and you need to manage expectations and also the environmental impact of some of the work we do. So I'd like to thank, I mean, there's a huge list of people I can thank, but everybody past and present in my lab, and also my collaborators, and thank you for your time.